So, welcome to my channel. In this video today, we will be talking about CIS and TRANS configuration. So, what do you mean by CIS? When two linked genes on each chromosome are of the same type, that means both dominant or both recessive, the arrangement is called the coupling or CIS. So, let's understand this one through a diagram. This is the diagram. Here we can see these are indicating the chromosome strand. These are two homologous chromosome actually, and in one chromosome if we observe, both the genes are in dominant form. Here also if we observe small a small b, that means both are representing in recessive form. That means in one chromosome, when the genes are located or present in a dominant form or in a recessive form, then such kind of arrangement is called the cis arrangement or cis configuration. On the other hand, if we talk about the trans configuration, so trans configuration means when two linked genes on each chromosome are of different types, that means one in dominant form and another in the recessive form. This kind of arrangement is known as trans or repulsion. So through a diagram, we will try to understand this. Here, this is again showing two chromosome strands, and in one chromosome, if we observe capital A, it is in dominant form, but in the same chromosome, if we observe in different loci, small b, which is in the recessive form. That means one in dominant form, another in the recessive form. But in this chromosome also same way, if we observe capital A, now it is in its recessive form small a, along with capital B, which is in dominant form present here. So that means in one chromosome, when one gene is dominant and another gene is recessive form, or one gene is recessive form or another gene is in the dominant form, then such kind of arrangement is mainly known as trans. Whatever the situation, cis or trans, but if we observe or if we talk about the recombination, so recombination between two linked genes occur with the same frequency, whether the alleles of the genes are in the trans or cis configuration. That means whenever the recombination happens in between the genes, then that time cis or trans configuration do not matter. Okay, so recombination, it happens in a same frequency wherever it is in the cis or in trans position. So this is about the cis and trans configuration of genes located on chromosomes.